Okay, let me call us to order. I hope everybody had a good lunch. We have an exciting afternoon. I'm going to introduce our first chair for today. It's Dr. Ted Spack, who we've known for a while. Ted has been in drug discovery and development for several decades now, and his primary efforts right now are as a managing director at Fast Forward, which is a nonprofit collaborative effort with Serono in developing drugs for multiple sclerosis, if I'm not mistaken.
Thank you, Howard, and I'd be remiss if I didn't say that it's an effort with the National Multiple Sclerosis Society, and this particular fund is with Serono, so it's an interesting public-private collaboration right now. Interesting. Find this slide. Oh, here we go. So, welcome back from lunch. Welcome to the Valley of Death. Nothing like a little death after lunch. The term Valley of Death gets bandied about quite a bit. Many of us use that term to describe that place where good ideas go to die, whether it's because of tox or PK problems or efficacy models that don't translate from animals to humans. It's a challenging area. Other people have referred to the Valley of Death as what happens in study sections. So that can also be a challenging area, but I think the real Valley of Death is where this session is placed. We're right between that post-lunch sugar crash and cocktail hour. So we have a challenge, but fortunately we have protection. We have the patronage and protection of the Roman god Janus, the two-faced god. Now, to look at him, you might think that he's the god of politicians. But actually, if you go to no less of an authority than Wikipedia, the source of all drug development knowledge, Wikipedia tells us that Janus is actually the god of beginnings and transitions. And so that seemed appropriate today. He's apparently the source of the words January, the transition from the old year to the new year, and janitor, which I haven't quite figured out yet. I guess the transition from a dirty floor to a clean floor. But he is, we're using him today to talk about how we're in this phase looking back and looking forward, which is something we always do in drug development. We begin with the end in mind. The clinical end is very important as we design even the earliest phases of the drug development pathway. And so you've heard in the morning a lot about high throughput screening, MedChem, ADME, PK. And what we'll be doing in this session is looking back to some of those elements and talking about how you integrate that into going into the clinic. With that, there'll be a series of talks which are actually interrelated. They go from What Makes a Clinical Candidate by Dave Weiner. Forgive me, Dave, I couldn't find your picture anywhere on the web, so this is the best I could do. But I share the group that he has brought his head today. And then I'll talk about requirements for an IND, filling in from Dave, taking a page from Dave and going further into that transition point between preclinical and clinical. Then we have Steve Parent talking about optimization and characterization of mouse models for neurodegeneration, how to use those and abuse them, and how you can use them to be predictive and what some of the caveats are. And finally, Barry Greenberg will talk about the very important topic of biomarkers, how they can be used in preclinical development, translatable endpoints. So with that, I'd like to welcome Dave Weiner to the podium, and thanks all for your attention to this session.
I'd like to thank the organizers and Ted for inviting me to this meeting. This is my first time at this meeting, and I'm very impressed. I thought the quality of the talks this morning was quite good, and I'm in a sort of transitional mode, as Ted had told you here. What I hope to do today is to talk about what you have to do when you're thinking about a clinical candidate from the view of a clinician. Okay, now this is not necessarily a treating physician who has a variety of different treatments that are available to treat patients that may come in front of them, but more from the highlight of a research physician who has to deal with a tremendous amount of uncertainty. So we have an unknown, clinically unknown compound or antibody or something for which we're going to try to translate that into a viable therapy. And that's a particularly difficult spot in the drug discovery spectrum. I would be very safe to say that pharma has done it quite poorly for many years now, okay? And this has now come to some realization as we've had multiple late-stage failures. And so the importance of this particular early transitioning into the clinic in the early stage is gaining tremendous importance as we move forward in drug discovery. And I think part of the talk today you'll see And what we hope to do eventually is have it be a seamless transition. So as I speak today, I'm going to do a couple of things in the next few minutes. I want to highlight some of the various factors that need to be considered in choosing a viable candidate for clinical consideration. What are the preclinical characteristics of a superstar, of a very good molecule to bring into the clinic? And how do you prepare for your first-in-human studies? I'm also going to talk on some of the objectives and the goals for really early clinical development. Not very little, if any, of my talk today will be about registration studies and NDAs. We're going to focus very much on that translational component. And then I would be remiss if I didn't talk a little bit about the goal. As Ted just said, at the end of the day, you have to be cognizant of what you're ultimately trying to address. And that has to be something you have to look at all stages of the process. So a little bit about the clinical dynamics of the treatment landscape, something evil called the TPP and time. That's what we'll talk about today. So drug discovery and therapeutic discovery in neurosciences and neuropsychiatric diseases is particularly tough compared to some of the other areas. And I remind everyone here that virtually all of the drugs approved in this space do not cure disease. They don't actually treat the underlying pathophysiological mechanisms of the disease. The best we've been able to do are symptomatic treatments, which are targeted to clinical symptoms or constellation of symptoms, which are the characteristics of those diseases. So many things are actually approved to treat the signs and symptoms of things like Parkinson's disease, etc. And so when you go down this difficult endeavor, you have to think about what is the ultimate goal, right? Oftentimes, it may be to improve upon existing therapeutic modalities where the current benefit-risk ratio is fairly favorable, right? So neurologic indications like Parkinson's disease and MS have multiple approved therapies where that has to be taken in consideration right above. You have to do something, you have to bring something to the table that is better than what exists currently or addresses an unmet medical need in those clinical indications. Or you may want to develop therapies for aspects of a particular disease or a disease itself that at present lack robustly approved therapies. I don't think I know any treating physicians who are really excited about acetylcholinesterases in Alzheimer's disease. I don't think riluzole is the end all for ALS, nor would anyone, tend to. So although there may be an approved product or two, there are many neurological disorders for which there is a profound unmet medical need. And that's an advantage as far as some of the principles we're going to touch upon that we were talked about this morning, like therapeutic indices, toxicology, etc. The Holy Grail are disease-modifying therapies, all right? I'm a partial optimist. I believe we'll eventually get there, but the challenges here are quite profound. That shouldn't stop us from trying. So I guess the first part of my talk, or the first question is, when do you get a clinician like me involved, all right? And then the answer to that is as early as possible, all right? I sit and I review many proposals, academic proposals, biotech proposals, et cetera, looking to bring things into the clinic, all right? And there's a couple of themes of things that are missing in those packages that are put together that we'll touch upon today that make them viable, attractive candidates, all right? And so what I like to think is, there's the actual selection of a target is a very complicated thing. But once you've selected a target, already you could start thinking about the biology, the physiology, and in our area, unfortunately, it's often not the pathophysiology of your target are actually going to dictate a lot of your later clinical planning and what you're going to look for, all right? And here you're a real estate agent. For me, the most important piece of data is where is your target expressed, all right? And the brain is a very unique organ because unlike the liver or the spleen, et cetera, there are subanatomical domains that are quite important and have quite predictive value with respect to treating particular symptoms or diseases, right? So already, very early in the process, years potentially before you're even going to have a clinical candidate, you can already be thinking about methodologies to assess the engagement of your target when you get to the clinic, all right? And this is a time to collaborate with others, to start thinking about things like imaging. And many of the larger pharma companies are, as they develop a small molecule for a target, in parallel are developing ligands only to be used as PET tracers for ligand-based imaging for later target engagement, all right? Functional imaging is really maturing to a point where you could start thinking about academic collaborations, and trying to interrogate targets of interest from functional areas, right? Physiologic, pharmacologic, one of the biggest problems here is that the brain is not a very accessible organ, all right? So a few years ago, even 10, 15 years ago, this was really difficult. But I'm quite optimistic, as a lot of these technologies improve, that at least for application in early clinical studies, these methodologies will help de-risk early clinical studies, will help identify which programs or which molecules within programs are worthy to bring forward, because you can validate the mechanism upon which you might be the hypothesis for why you're trying to spend effort on a particular target. All right? In the talk this morning, it was very interesting to see that even the HTS methodologies are starting to incorporate very early on things like patch clamping, electrophysiology, and it would be great to think of the seamlessness of that all the way through the drug discovery process if possible. All right? This also, your location also can guide your target organ toxicological assessments. And you heard a very nice talk this morning about toxicology, where we look everywhere, but you look extra hard in places where your target itself may be mediated, because a simple view from a clinician on toxicology is there's toxicology related to your target and mechanism, and then there's all the other stuff that isn't related to it. And at least you get some handle on that which may be target mediated. All right? And in general, you can even think about adverse effect profiles and tailored safety assessments. Right? So if your target is expressed in the eye, you've got to start worrying about the eye early. Right? We're going to hear a little more about cardiovascular. And if your target is not expressed in the testes, I'm not worried about target-mediated testicular effects. See, so you can start planning quite early. Pathophysiology, as I said, is a difficult one. But as we move forward into targets that are genetically defined, that may have a pathophysiological role, you're already thinking about patient populations, right? So one of the things, and I work with the Michael J. Fox Foundation, as we move LRRK2 molecules forward, we're already planning years in advance to enrich that subpopulation, to go out there and study cohorts of people with those mutations, because that's where those compounds are going to be tested. And so you can't start even early enough, as you can imagine, in trying to think about who are we going to test these compounds, how are we going to show that they're effective at a later point. And once again, the development of objective biological measures or biomarkers, and there will be an entire talk about that later today, or novel, and they're usually subjective clinical outcome measures. All right? So for this group, I thought I'd pull out MS. And when I think about the pathophysiology of MS or any of the neurologic disorders, I just am impressed over the years about how little we know about neurologic disease. All right? We think we know much. We think we can characterize much at many levels. But at the end of the day, what we don't know far exceeds what we know. And one of the points is, as you go through a drug discovery process, the science changes, right? So those of you for Alzheimer's who weren't Taoists maybe are now, and so things change over time, and you have to keep that in mind as you're bringing things forward, and really have some hubris about what you think we may know or we may not know. So here was from a review from a few years ago of various targets in MS. And one of the unique things about MS, why it has been so successful in the drug discovery realm versus something like Alzheimer's, et cetera, is that many of the targets are peripheral. So we don't have to worry about trying to get our drugs into the CNS and balancing that with peripheral side effects. And it's immunologic, so very generalized immunosuppressants have efficacy in MS. And we also have a biological marker, and that's structural MRIs are quite sensitive to pick up anti-inflammatories. So that helped stack the deck and led to a whole series of successes in the MS realm. But as these immunosuppressants become, and there's still room to get more selective agents based on the autoimmune processes selective in MS, they're now shifting much more towards things on the other side of the brain barrier and neuroprotection. It's going to get a lot tougher, because now the people working in MS are going to be in the same difficult situation that a lot of us are that work in other neurodegenerative disorders and trying to understand neurodegeneration and address the appropriate targets in a clinical means. All right? It's always nice if you have a team and you have an early clinician or somebody that's thinking about one of the key steps where you really start to get involved is lead optimization. And it was a very nice talk this morning about some of the various aspects. And this is critical, because another fundamental take home from my report is I would much rather have a molecule that has really good pharmaceutical characteristics and may address a target that may not be that sexy versus a sexy target and a terrible molecule. Right? And we have brought many, many terrible molecules forward for sexy targets, and they fail. And then we don't understand if that failure was actually related to engagement of that target, or whether it was just the fact that we couldn't get enough of it there, we couldn't get a wide enough therapeutic area. So for me, as I look at opportunities, the ones I like the best are the ones that have the best characteristics to be able to give the most leeway in the clinic to actually interrogate the question, right? So I agree that I think it's very, very important that optimization efforts, take your best and brightest forward during the lead optimization, is critical, right? give up absolute potency for better metabolism, all right? Maybe give up some relative selectivity as you work through it. It's a difficult process. The chemist this morning did mention when you tinker with one, it affects many of the different variables. But this is a stage where I can't underestimate how important it is to have what comes out of this be as optimized as possible, because that's really, later on, that really comes back to bite you. All right, and so in the ADME or ADMET, you know, as I mentioned, sacrifice some of these issues, and PK can't be underestimated. I mean, this happens to be a PK profile of a compound in phase 3, and what's really nice about it is, look, a very well-defined, you know, steady-state doses, clinical dosing differentiated on a PK perspective, metabolites that are measurable and quantifiable and in relative work. This is a dream for clinicians that bring things into the door. And in this particular molecule, it enriches in CNS versus plasma. And that's also a very, very important component. That those are the molecules I look for, because we can design clinical experiments that test hypotheses and ultimately, hopefully, can move things forward. All right? As you're moving through the preclinical process, you've got this animal to human transition. All right? And here, I apologize to the speaker, Steve, that's following me, and any behaviorists in the room, but here is where I think we've run amiss with efficacy models in neurologic disease. They are not predictive at present. We need to put more effort into them. They're valuable, but at present, I wonder how much effort in decision-making we use in moving those forward and whether that's valuable. Preclinical safety assessments are requirements for INDs. Keep in mind, almost everything we treat is chronic dosing, all right? And the point made earlier about, you know, having toxicology to cover the amount of time in the clinic, that's something that evolves over time, but keep that in mind, right? You're not going to treat patients. You're not going to do an initial clinical study often with one dose. It's going to require multiple doses, and you're going to build from there. So you've got a plan for chronic dosing. Route of administration, very important. We all think about oral, we think about IV, but in CNS, where there's also intrathecal, and there's also intraventricular. And I've had the privilege to work with companies that are doing intraparenchymal neurosurgical stereotactic injection of growth factors, you know, for Parkinson's disease. I mean, these are big, big challenges, but hopefully the clinical outcomes are going to be quite large as well, all right? And things like treatment modalities like deep brain stimulation have opened up the technologies, the methodologies, and the safe use of even intraparenchymal injections. But I do caution you that that's a very tall bar to overcome, all right? Determination of therapeutic index, all right? Now, let's pause here for a moment. Therapeutic index can mean many things to different people. Ultimately, at the end of the day, when you're filing your NDA, it's efficacy versus adverse effects. But you can think about therapeutic indices at all steps along the process, all right? And like, I know there was some discussion this morning, somebody wanted to quantitate it. I say, go wide, you know what I mean? I like to see compounds 10x, 20, 30x, and the toxicologist mentioned this morning that it rarely gets wider. It always seems to get smaller over time. And that's probably very true in the clinical arena too, because we can't predict everything. So the wider the therapeutic index for whatever your measure of efficacy may be, it may just be a pharmacological effect early on or a pharmacodynamic effect, but that versus any toxicological or adverse findings, it's very important to choose candidates that have very, very wide therapeutic indices, all right? I mentioned often here the optimization for ADMET. I cannot agree enough with the discussion this morning about characterizing early brain penetrance, but not only penetrance, but the actual residence time, how well your molecule gets into the CNS. Please go out and get more LCMSs. I see so many grants, so many proposals where that's not been well characterized. And not only does it hold things up, but many things die along the way, because either things were dosed inappropriately in models where the blood-brain barrier is broken down, where there's not very good quantification. I can't emphasize enough how important that is, and that's a recurrent theme in this field about characterizing that well, all right? Preclinical efficacy models, they are not required for an IND. You can go all the way to the clinic and get a drug approved without ever curing a rat of Alzheimer's, all right, or Parkinson's or any of these. And people oftentimes don't realize that. And part of the problem here is because they often lack validity or are biased towards specific mechanisms, right? You know, my biggest concern is that we're actually passing on things that don't read out in these models that could be viable and effective therapies. So if you have a hypothesis, if you're approaching a system, you're approaching a target, and you believe in that target, don't necessarily stop because you couldn't demonstrate efficacy in a non-predictive animal model, all right? And I wonder if we're missing gems. But what are very important in the use of the animal models, even the existing ones now, are to demonstrate a meaningful effect of modulating the target biology that you have, right? So you can use these models to show pharmacologic effects, pharmacodynamic effects, or biological effects, and quantitate the exposures needed to do that, and that is very valuable information. And those should be go, no-goes as you move forward. All right? So these are very, very good areas to do PK/PD marker development, to translate methodologies, right? So, you know, if you're doing certain interrogations in animal models, be it electrophysiology, optical coherence tomography, there's functional imaging even now, or as well in animals, you can translate that into the clinic. So work done here in those animal models are very, very important. And my one view on biomarker development is start early and collaborate widely, because no one person can do this. This requires multiple people at the table to not only propose a biomarker, but to bring one forward that's viable and could be validated. When you're at the preclinical thing, how do we optimally predict that human dosing will be safe? The answer is we really can't, but we've done actually quite well as an industry over time, right? Safety pharmacology, specialized studies to define, as I mentioned, both the known, the cardiac small molecules, a very good example, and anticipated, you know, dopamine agonists in the Parkinsonian field have ocular effects. You know, the secretase inhibitors and dermatologic effects. There's certain things that you can, as you learn more about the biology and within the safety pharmacology, interrogate. All of that information critically important for clinicians as we try to choose dose and design early clinical studies. Toxicology, just heard a very nice talk. The key point there is toxicokinetics. I see many, many early tox programs without exposure, and they're not very helpful because you know, without understanding what types of exposure you're getting to see the effects, they become of limited value. And we already talked a little bit about the length of exposure being critical. Okay? Now, estimation of starting dose. There was a question today about isometric scaling. I'm not going to speak specifically about that. There are existing guidelines, but the general principle is that exposures that you've got in animals that have led to NOELs, or no effect doses versus MTD doses, are then scaled up based on existing formulas to choose a starting dose in humans. And there's two things here. One is try not to be too conservative, because it's very, very difficult and it's very expensive in humans to try to, you know, through cohorts of multiple people do PK if your starting dose is far, far below your pharmacologically active dose. But also, it depends on what species you do your scaling up on, because, you know, the metabolism, the distribution and the ADME of drugs can differ very much between species. And so you'll have isometric scaling and starting doses from your monkey studies that may differ from your dog or your rodent studies. And so obviously at the end of the day, this is a discussion between regulators and yourself as to where the starting dose should be. Regulatory. Interaction should be early and often. It could be summarized basically there. Consider regulators as your partners in development. Remember, they see everything from every sponsor in a particular indication, and oftentimes multiple you know, submissions around a similar target. So they can educate you as you go forward, and they should be partners of yours in the process. And I'm a strong advocate for pre-IND meetings. Okay, highly recommended for novel targets, novel indications. You know, cognitive impairment in Parkinson's disease, et cetera, and or for INDs from academic groups or small sponsors. All right, Pfizer does not get granted very many pre-IND meetings, right? But obviously, many people in this audience, you should seek it. Nothing lost by requesting it, but the key is you have to have a specific question, right? Regulators are quite busy, right? So you've got to have a question you're thinking about that you want to get answered. Often it involves the early clinical development plan or specialized safety monitoring in early clinical trials. Clinical trial design, all right? Have a CDP. So, you know, even though you're ending somewhere, you want to end it with a vision of the molecule, right? And in order to get to that profile, you're going to have a plan, and that plan will evolve over time. But you should already be thinking about the early clinical aspects while you're still in the preclinical setting. So, your aspirational goals will yield a product profile. You know, you want to confirm mechanisms related to the biology and the pharmacology in early studies. All right, now there's more methodologies available to do that. And you want to explore the relevance of this in multiple populations. All right, so one of the mistakes we have made is we've buttonholed things. We said this is a treatment for disease X, and we never explore it when it gets to the clinic in variants therein. So maybe it's for Parkinson's, but we never explore PSP or MSA or any of these other, where there could be even a stronger rationale and a larger clinical effect size in those smaller indications. All right? So pharma is moving towards these orphan diseases or niche indications. That should be applied in this drug discovery and neurologic disorders. And the core elements of any clinical trial, who are you going to test it on? Patient population, the starting dose, the route of administration, the dosing intervals, and the duration. Right? So we heard earlier this morning, dose is not just 5 milligrams, it's much more than that. What safety assessments, the timing of them, right? We do cardiovascular assessments at Cmax, not at Cmin, right? The timing of them, and PK evaluations, right? CROs, and I apologize, make a lot of money on PK assessments in early clinical trials. You don't have to do the Cadillac of PK assessments in every trial, all right? There's a time and a place for all of that, and much of it is in the early setting. And try not to accomplish too much in a single clinical trial. All right? I've seen many of them fail because the actual investigators cannot do all the things you've built into a trial. It looks great on paper, but you can't actually pull it off because you've front-loaded it with so much that neither the investigators nor the patients can deliver reliable data. All right, I'm going to go over just a minute or two if that's okay with that. First in human, this early phase 1b, safety and tolerability and early indications of TIs. Generally, for me, it's not clinical outcome measures, although we do use things like UPDRS to make sure that we don't worsen motor function in Parkinson's disease, maybe visual analog scales and pain for early assessments of analgesic activity. But in general, these are safety measures and targeted clinical scales. Strong consideration for first-in-human studies to be done in patients, not normal healthy volunteers. This is the cancer model, but I would argue strongly that for most neurologic disorders, because of the neurodegenerative process itself, because of the age of the patients, the safety and tolerability sometimes differ between that population and a normal healthy volunteer population. And the earlier you get into patients, the better. All right? This is the time to characterize the pharmacologic effect, because at phase 3, you can't do that as well. All right? So interrogations to look at putative biomarkers, please do them early on. This is not a regulatory study. Explore that. Do science. Do clinical pharmacological experiments in your early clinical studies. Lymphocyte dynamics in MS. There's a variety of successful approaches here to confirm that you're engaging your target and that engagement may lead to a physiologic effect that you predicted and hope will be therapeutically relevant at later stages. And you can also plan and initiate special studies like imaging, pharmacodynamic, and some of the other phase 1 studies you need to do to round out an eventual NDA package. Many of these are related to safety and drug disposition. All right? As you get later on into the process, into the phase 1b, flexibility to add additional cohorts and adaptive trial designs or small dedicated studies to answer scientific questions don't happen often enough. Don't be afraid to do those, all right? They're hard to argue for sometimes, but they're very, very valuable, all right? And like I mentioned before, you want to explore various populations and disease indications if you have the time and the money and the right partner to do that. As you know, phase 2 studies, you're doing proof of concept here. Your primaries have to be clinically relevant measures that you're going to carry forward into phase 3. But you have some room in the secondary area here to look for biologically relevant measures. And you already have to start thinking about phase 3 and beyond and incorporating things like quality of life, health economic, and if you are working in a space where there are others in that target, clinical differentiating measures. All right? But the size of these studies in general are not powered well enough to really interrogate these measures. That has to wait for phase 3. And as I mentioned, the registration studies, we won't talk about them today, but they're there to confirm efficacy and to really round out the profile that you have. And I'm going to end today on and just in that comment, here's a representation of some of the drugs that are in development for Parkinson's disease now. And you can see down here the marketed drugs, just how many there are, all within a dopaminergic realm, and how many are coming down the pike. So you have to think about how you're going to differentiate yourself. If you're going to have a clinical effect on neurodegeneration and stop the disease progression, there's no differentiation. You've hit a home run. But many times there are incremental advances in therapeutic development. You have to appreciate the landscape. You've got to understand where you're going to help design the earlier studies. What I thought I would do was hopefully I can come back to this conference a few years from now, and this will be the drug that somebody in this room discovered. It's quite a good drug. We'll call it Amritra. It's indicated not only for the treatment of Alzheimer's because it stops the disease in its track, but it also prevents Alzheimer's. All right? And so you can take 1 tablet by mouth daily. It doesn't matter with or without food. There are almost no side effects. We get a little bit of headache, maybe some flatulence, but very few side effects. No drug-drug interactions. and there's no medical contraindication to use, all right? So how did we get to this drug? Well, the take home here was the preclinical characteristics that allowed the clinical developers to get there are certain types of characteristics. Highly potent and selective inhibitor of a novel target that was only expressed in the CNS. Its GMP manufacturing was quite simple. There was low cost of goods. They didn't encounter any impurities. It was stable at room temperature with a shelf life of over 3 or 4 years. Reversible toxicological findings. One of the worst things you can have in toxicology is no findings at all, right? So you want to have a toxicological signal, but you want it to be something that's reversible once you've withdrawn your therapeutic. And it had that, but exposures 50-fold greater than the therapeutic exposures. Its ultimate safety TI was over 100. 90% oral bioavailability, linear pharmacokinetics, a half-life of 16 hours to support once-a-day dosing, no accumulation, and is excreted essentially unchanged in urine and had a 10x plasma to brain-to-plasma ratio. And lastly, the target itself had no polymorphism in the population, so everyone responded, okay? No genetic or pharmacologic modifiers, and nearly 100% response rate with no tolerance over time. And to get there, you need 2 things. You need to be persistent, and you need some serendipity, and I wish all of you in the audience plenty of the latter. Thank you.
Thank you for a great talk. So you raised an issue which I think has really been battered around a lot lately, which is the whole issue of transgenic animals as a barrier in a way, rather than as an accelerator, let's say, of drug discovery and things going into the clinic. And I think you raised it in an interesting way, really, because you offered an alternative, which was to use animals for pharmacology, I guess, and pathophysiology, but not for these target-based. So we say in Alzheimer's we've cured mice 350 times, but yet, I think the vast bulk of research that I know of, both in industry and in academia, relies on these models. And so 2 things. One is, if you could elaborate more on why, how we get around the models, let's say, if that's a good thing to do, and arguably may or may not be. But I think more positively, like in the context of let's say Alzheimer's or Parkinson's, where you really don't know the mechanisms, how would you go at what you were saying in your second point on that, which was testing the testing potential clinical candidates in pharmacology and pathophysiology, and I guess in the normal animals, what you were saying, really a normal.
Well, or it could be but see, this is part of the problem that makes it so difficult in this space, is that funding decisions, late development, even POC studies, are quite expensive, right? The amounts of money spent there far exceed, some of the amounts spent to get there. And many senior executives in pharmaceutical companies at least believe or feel more comfortable if you've got animal efficacy to bring that forward, all right? And I think it's something we have to move away from. I don't have an absolute answer, but I think we just have to learn from experience. And one of the things is we've been failing miserably. We have to have a plan B, all right? And so my suggestion to an audience like this is what could be equally compelling for somebody like me is to understand the biology of the target. So this is academic investigations to understand the role of that target, the physiology demonstrating that your therapeutic engages that and goes down that path, rather than the absolute piece of evidence that let's say something like autophagy, right, was able to lead to a improvement in cognition in an animal model, right? Now, what you may see in that animal model is you may clear a species of a complex protein. We're not even sure which species we want. I mean, the uncertainty is tremendous. The positive data makes us feel better, right? But I'm arguing at every level that we have to somehow move past that and do science and test these hypotheses in patients, and ideally to move that bar, and that's really a target validation bar, from an animal into an early human setting, right? So if you can go into a patient population and show based on A beta imaging, that you can decrease the levels, that's going to give you the reason to go and invest in that therapy and potentially go to phase 3 and to move on. So I'm suggesting there will always be a role for this, but that we have to come to a conclusion that until these models are improved, until they get to a better place, right, we have to be willing to continue to invest in novel therapeutics that lack that box on the checklist that you have for, and to move the bar a little bit further. And so instead of thinking about an animal model, think about the methodologies, because we can't get brain tissue in human studies, about how you're going to measure that in a human. I'd rather see people spend more time thinking about that and demonstrating pharmacodynamic or pharmacologic effects in a human population, rather than a behavioral or other outcome in an animal model, which may not be predictive of efficacy.
I sort of agree with you, but it just seems like it ain't going to happen for some reason. Like because the safety toxicology, IND enabling phase 1 stuff, it's millions of dollars, right?
Yeah.
And before you finally get to that phase 2A where you finally ask the question in humans, and if you try to, even through our foundation, I would say,
It's
or
tough to do.
especially in industry, try to get into all that process without having an effect in an animal model, it seems like.
Yeah. Well, I suggest, there's a tremendous unmet medical need for many of these neurological, and the pipelines, the early pipelines for most major pharma, I know them well, are not chock full. That I think there will also be a certain bit of starvation-induced willingness to maybe move forward with things that are beyond those that we but that's a very complex thing to answer. We'll have to see. If I couldn't have predicted the successes in oncologic drug discovery 15 years ago either, but things change over time.
So, David, I guess I wanted to respond to the sort of the last comment there. I do think that there would be appetite to do that, and I think it's a question of how you construct the package that you bring forward. I sort of think about it in the context of the decision makers in a pharma environment is really about it's about mitigating risk. It's about managing risk, and the question is, does the asset that you're proposing to put more investment in, what is the risk that it carries going forward? And can you minimize that? And if you've done a non-predictive animal model in the best way possible, you haven't mitigated the risk necessarily of it failing downstream. But if you've conducted a mechanistic study in a small subset of patients appropriately stratified so that you can actually measure the effect in a meaningful way, that's going to change the risk profile of that asset, and you can then take on the multitude of millions of dollars that you might need to do the powered studies required from the regulatory perspective. So I guess maybe a different way of